Hello there. Here's what I found.
Neurulation is the process of forming the neural tube, which will become the brain and spinal cord. In humans, it begins in the third week after fertilization, and requires that the neural folds, which consist of the ectoderm, come together and fuse in the midline. During neural tube closure, hinge points display dynamic and polarized cell behaviors necessary for converting the neural plate into a neural tube. At hinge points, polarized cell shape changes result in cells with reduced apical and increased basal cell diameters. Bone morphogenic protein, aka BMP signaling, is essential for proper hinge formation. BMPs signal via the canonical, which is SMAD-dependent pathway, or non-canonical pathways. In the canonical pathway, BMPs initiate the signal transduction cascade by binding to type 1 or type 2 serine threonine kinase receptors and forming a heterotetrameric complex. The constitutively active type 2 receptor transphosphorylates the type 1 receptor at a glycine-serine-rich motif, which is the GS domain. And then, activated type 1 receptor immediately phosphorylates the receptor-regulated SMAD1, 5, 8, aka RSMADs. Phosphorylated RSMADs associates with the co-mediator SMAD4, which is called co-SMAD. And this complex translocates to the nucleus, where it functions as a transcription factor to regulate gene expression. As mentioned earlier, BMP signaling plays an essential role in neural tube closure. BMP inhibits MHP and DLHP formation, whereas repression of BMP by an inhibitor allows DLHPs to form. In neural plate cells, PAR complex localizes at apical junctions, whereas the lethal giant larvae, aka LGL complex, is associated with the basolateral cell membrane. In the presence of canonical BMP signaling, phosphorylated SMAD5, 8, recruit PAR complex that stabilize junctional proteins on the apical surface, and LGL is restricted to the basolateral membrane. Therefore, the size equality between the apical and basal membranes are maintained, and hinge points are not created. However, noggin can weaken the BMP signaling by blocking BMP ligand from binding to its receptor. It leads to reduced phosphorylation of SMAD5, 8, so the PAR complex becomes unstable and disrupts the tight junction, resulting in apical constriction. So, for the proper formation of hinge points, the correct amount of both BMP and noggin are important. Defects of neurulation result in clinically important congenital malformation, termed neural tube defects. Failure of brain closure and low spinal closure occur with a high prevalence in humans. Therefore, the precise control of BMP signaling is essential for preventing neural tube defects as well as proper hinge point formation.
You're welcome. Goodbye.